Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. Last video, we finished up Chapter 8 in our discussion of cell division. This video, we're going to be beginning our march into Chapter 9 and our exploration of genetics. So to begin with, we're going to start out at the very beginning and start talking about Mendel's laws with the first up, law of segregation. As always, before we begin, right now you should see our terms and definitions slides popping up so you can grab the vocabulary that we will be using in today's video. A quick note, this video and the next one will have the same terms and definition slides because we're going to be using the same vocabulary for the most part. While we may not get to all the terms at the end of these slides in this video, we will be discussing them in the next video. And I chose to break up these two videos while I was making the PowerPoint, so it made more sense to leave all the terms and definitions in both PowerPoints. Also, if you don't want to pause the video and grab the terms and definitions, a quick reminder, you can get the PDF of my slideshow for absolutely free on my Patreon, which is linked down in the description below, as well as any other study material that I have made to go alongside these videos. Also, please don't forget to like, comment, subscribe if you like these videos, if you're finding any use in them. Is my goal for them to help out anyone who may be struggling in an entry-level bio class, and it really does mean a lot to me when I get feedback on that. So without further ado, let's jump into the law of segregation and begin our discussion of genetics. Before we jump into the actual science and nitty-gritty biology of this field of genetics and the experiment done that laid the groundwork for the field, we need to have a little bit of a history lesson because it's important to understand where the ideas that brought the field to where it is today came from. So this whole study of heredity, study of genetics, thinking about how things get passed on from one generation to the next, has very ancient roots, specifically way back to ancient Greece with the physician Hippocrates. This physician had the idea that particles that he coined pangenes would travel from different parts of the body to the sperm and the eggs. Now, the language used would not have been sperm and eggs. They did not have those terms back then, but that's the basic idea, that the things that happened in your body, from like arms, fingers, hair, whatever, when it came time to make the next generation would kind of breed these little particles that would all get mixed together and pass on. Now, this is ultimately wrong, and we know this today, that gametes are not composed of particles from somatic cells. So the egg and the sperm, the cells, the sex cells that pass on genetic information to the next generation, they are not made of a cell from your eye. The little bits and pieces do not travel into the sperm and the eggs. We know about the process of meiosis. We went into that in Chapter 8. If you have not seen that video, please go back and do so. Later in this video, we will touch on topics from that one. But we also know that changes in these somatic cells, so the cells that make up the rest of you that are not part of the gametes, they don't really affect the gametes. Now, put a little pin in there. There is an entire field of genetics that does look into how the environmental changes can affect offspring down the line in future generations, but in general, we are not talking about that. In general, the changes in your somatic cells do not affect the gametes. So this whole idea of the pangenes is ultimately wrong, and we do know that now, and we do know why that is wrong now. So after the ancient Greeks, there wasn't a whole lot of change in the ideas behind how things get passed on. So we're going to fast forward here up to the early 19th century. And in the early 19th century, the whole idea that kind of populated this idea of heredity and how traits get passed on was the blending hypothesis. Essentially, what the blending hypothesis says is that all this genetic material from mother and father will mix in the offspring, sort of like paint. So if you have like a yellow paint and a blue paint and you mix them, you're going to get a green pigment somewhere in the middle. And that was essentially the hypothesis that dominated in the early 19th century. One example of kind of how they thought it would happen in a biological setting is if you were, say, breeding black labs and brown labs. Now, if you were to breed a black lab to a brown lab based on this hypothesis, what you would expect to see is that there would be a blending of those coat colors that would be somewhere in the middle of the black and brown. But is that what you actually see in the puppies? No. What you're actually going to see if you breed that purebred black lab to that purebred brown lab is that all of the offspring are going to be black. So what exactly is going on? Why aren't the colors blending? Well, we're going to bring that up. We're going to kind of answer that question as we move into what Mendel actually did and how the groundwork of the field of genetics was ultimately laid. Gregory Mendel is the main character in this whole tale of how the field and study of genetics had the groundwork ultimately laid and the whole field of study came to be as it is today. So he was an Augustinian monk that lived and worked in an abbey in what was at the time Brunn, Austria. Prior to studying heritability by breeding pea plants, he studied physics, mathematics, and chemistry at the University of Vienna. And it was in 1866 that his revolutionary paper was published, and the field of genetics was kind of born. The big ideas from this paper was that organisms have heritable factors. These heritable factors retain their original identities from one generation to the next, and the heritable factors can be shuffled but do not blend. And we now know these heritable factors to be genes. So while his language was heritable factors, they didn't know what genes were at the time. We now know that they're genes. And this whole idea of genes being shuffled but not blending and being these little discrete things that maintain their own identity from one generation to the next was very new. Again, the most popular hypothesis at the time was that blending hypothesis. But we now know the blending hypothesis is not the best description of what goes on. So why did Mendel choose to use pea plants instead of something like dogs, for example? Well, pea plants have a very short generation time, so he didn't have to wait years between breeding events, and he was able to conduct his experiments in a relatively short period of time. If he had used something like the black and brown labs that we used in the previous example, it would have been a couple of years between each generation, and the experiment would have drawn out for a very long time. Also, pea plants have a lot of offspring per mating event, so he had a very large sample size to work with when he was working with pea plants versus things like dogs that have a much smaller sample size. And in science, you want larger sample sizes so that the kind of noise and the anomalies kind of get worked out on their own, and you get a more clear picture of what's going on. And also with the pea plants, there were already quite a few varieties that were easy to obtain, but were also easy to distinguish from one another, which was very important because at the time, they didn't have the technology to just kind of look at the genome and see what base pairs were going to be kind of together and what was changing from one generation to the next at that molecular level. They were basically limited by what they could actually see with the naked eye. So these easy to distinguish pea plants made it very convenient. So what exactly was Mendel looking at? Well, he was looking at characters and traits. And let's not get too hung up on this terminology. So the specific heritable features that vary among the individual is going to be the characters. So things like the flower color, flower position, seed color, seed shape, pod color, and stem length, like you can see in this table, are the characters. And these were the characters that Mendel was actually looking at. In this video and the next one, we're only going to be discussing a couple of them because ultimately, if you can understand it from what we discuss, you've got a good grasp on the basics of genetics and the whole Mendelian genetics thing. So notice how these characters have two versions each. Well, these different versions are the trait. So character is the overarching thing, the flower color, for example, and the trait is the individual variant. So the purple flower and the white flower are the traits. So what exactly was the experiment? How did it work? Well, the first thing Mendel had to do was choose these true breeding varieties. Now, with the pea plant, a true breeding variety is when you use that plant to pollinate itself, so self-pollination, and the result is 100% offspring identical to the parent. So you take a purple flower plant, make it pollinate itself, and all the plants that result are purple flower plants. That would be a true breeding purple flower plant. So then he used some selective breeding techniques to then see what happened in the offspring. So first, when he was actually crossbreeding the plants, he had to prevent self-pollination. He did this by removing the stamens, the parts that produce the pollen, so you can't accidentally self-pollinate, and then just manually pollinated. So take a little brush, get some pollen, and kind of slather it onto the part of the plant that accepts the pollen. We're not going to get very into the parts of the flower here. We will discuss that in later videos when we talk about plants. So don't worry about that terminology in the table here. Just know that to prevent self-pollination, he removed stamens and then manually pollinated with like a little brush. And then in some cases, he promoted self-pollination by covering the flowers of the plants with little bags. And by doing that, it just keeps pollen from other plants from getting in. So the only way that plant can be pollinated is through self-pollination. And by using these techniques, what he did was he crossbred those true breeding plants. So he crossbred the purple flower plant with the white flower plant. So the resulting generation were what we know as hybrids. And once the new generation was here, he observed the characters and the traits in the offspring. So that very first group of offspring is known as the F1 generation. The F1 generation is just a way that we know this was the first generation. This was the first thing that was produced through this experiment. And then once he had this F1 generation, he bred those hybrids, primarily through self-pollination, but there was some cross-pollination, but that's not really the important part here. The F1 generation, the interesting things came through the self-pollination technique. And then he observed the characters and traits that popped up in that F2 generation. So F1 and F2 are just how we kind of distinguish which generation came first. The very first generation, the parental generation that kicked the whole experiment off, is known as the P generation. Again, you don't have to get too worried about the specifics of these terms. Just know that it goes P generation, F1, F2, etc. That's just how we denote which generation is which in these heritability and genetics experiments. So what were the results of Mendel's experiment? A quick recap, this P generation was the purple and white flower plants, and they were the true breeding plants. And so he crossed them. He made this hybrid F1 generation. A hybrid is just something that is a cross between two things. And what he saw in this hybrid F1 generation was all purple flower plants, all of them. So what happened to the white flowers? Was that trait lost? Well, the only way to know for sure would be to self-pollinate and make that F2 generation. And what he saw when that happened was that for every 3 purple flower plants, there was 1 white flower plant. Now, if you want the exact numbers, they are out there. You can find them. If you look in this book that I'm using, it gives the exact numbers, but it's not really too important to get the basics. So the big thing here was that the white flower plant existed in this F2 generation. So the white trait was not lost. But what the heck is going on? What does this all mean? Why was the F1 generation all purple flower plants, and why was the F2 generation 3-to-1 purple white? What's going on? So the explanation here kind of has these 4 hypotheses that go with it. And we're going to be using the modern language, not the language that he would have used with heritable factor, blah, blah, blah. We're going to be using genes, alleles, all of those more modern terminology. So the first hypothesis is that there are alternative versions of genes that account for character variation. So the alternate versions of these genes are called alleles. So the gene is the flower-colored gene. The allele is the allele for the purple or the white flower. Hypothesis 2. For each character, an organism inherits 2 alleles of a gene, 1 from each parent. And then a homozygous individual is going to have 2 identical alleles of that gene. So the example would be 2 copies of the purple allele. And then a heterozygous individual has 2 different alleles of the gene in question. So 1 white flower allele and 1 purple flower allele. Homozygous, 2 identical alleles. Heterozygous, 2 different alleles. Hypothesis 3. If the 2 alleles of an organism differ, then one determines the organism's appearance and is called the dominant allele. The other has no noticeable effect and is called the recessive allele. So dominant alleles are going to be represented when you're kind of writing things out and in diagram form with a capital letter. In this case, in this table, in this diagram, the purple allele is the dominant allele and is denoted with the capital P. And then the recessive alleles are going to be represented with a lowercase letter. So the white allele is the recessive allele and is denoted with the lowercase p. In a little bit more simple terms, what we have going on here is a dominant allele is one that kind of covers up that recessive allele. So if you have 1 purple allele and 1 white allele, the flower is always going to be purple because purple is dominant to white. Purple covers up white. The only way to get the white flower is to have 2 copies of that white allele. So the individual would have to be homozygous for the white allele. And then hypothesis number 4, a sperm or egg carries only 1 allele for each inherited character because allele pairs separate or segregate from each other during the production of gametes. Again, go back to our meiosis video. You'll see the homologous chromosomes separating. That's when it happens. That's when the allele pairs separate. And then this is also known as the law of segregation, and it's the big thing that explains this observed inheritance pattern. So a quick review here before we move on to a couple of examples and a little bit more talking about the diagram that we saw on the table is that phenotype is what you see. So phenotype is what's visible on the outside, and genotype is the genetic makeup or what alleles are there. Then a gene is found on a particular locus on a chromosome, and homologous chromosomes carry the same gene on the same locus, but they may have different alleles. And I know that's a whole lot of words to take in, but this slide is all review from previous videos. And the little image we have here are two homologous chromosomes. You can see the different loci on the chromosomes here, and you can see the genes that are represented. And for the most part, this particular homologous chromosome pair, this organism, is for the most part going to be homozygous for the P trait, the A trait. However, if you notice that last little locus there, that last gene, this individual is heterozygous for that B trait. So homozygous for the dominant allele at that P locus, homozygous for the recessive allele at that A locus, and then heterozygous for the B allele at that B locus. So the homologous chromosomes have the same genes, but they may not have the same version of the gene. And that's really the big picture there. Because one chromosome came from mom, one from dad. Mom doesn't necessarily have the same versions of the gene as dad does. So that's why we get the possibility of the alternate versions of the gene. So let's talk a little bit about that diagram we saw, that diagram that lives in the nightmares of biology students everywhere, the Punnett Square. Now, I actually like Punnett Squares. I actually really like making them, so I apologize if I get a little too excited here. And basically what the Punnett Square is just a tool to visualize the possible offspring in a cross. So this one from the book is the Punnett Square for that flower color. So what we see here on the little side is the phenotypic and the genotypic ratio, because there's a difference in what we see versus what's going on in the genome of the individuals. So on top, we see the two alleles from the sperm. So we get the big P, the dominant purple allele, and the little p, the recessive white allele. And that's going to be the same for the egg from that F1, from the F1 plant as well, because you have a 50-50 shot. The plant can pass on either the dominant gene or the recessive gene. Excuse me, the dominant allele or the recessive allele for that gene. And what we see here are the 3 purple plants and the 1 white plant. But if you look at the actual alleles assigned, in one square, you get the 2 capital P's, you get the homozygous dominant purple plant. So that offspring, that possible offspring, is a purple plant, but is homozygous for that dominant allele. And then if you move 1 square to the right on the top, you'll notice it's another purple plant. However, in this case, the egg passed on that dominant allele, but the sperm was carrying that recessive allele. So in this square, we have a purple plant that is heterozygous for that flower colored gene. It has 1 dominant purple allele and 1 recessive white allele. Now, if we move down to the bottom row, we can see the same thing in that bottom left square, except for, in this case, the dominant allele came from the sperm and the recessive allele came from the egg. But if we move over to that very last square, that bottom square on the right, we see that this flower is going to be white. The egg and the sperm both carried in this hypothetical offspring that recessive allele. Therefore, it's homozygous for the recessive gene and is going to be white. So the differences that we see here, the phenotypic ratio, what we see is 3 purple, 1 white. But the genotypic ratio, what happens at that gene level, what we don't see is that we get 1 homozygous dominant, 2 heterozygous plants, and 1 homozygous recessive. So the phenotypic ratio is not necessarily going to be the same as the genotypic ratio. We will get into that a little bit more in the next video. But that's kind of the groundwork there to kind of understand that what you see isn't always what's going on in the genome. So the phenotype does not always equal the genotype. The phenotype is going to be what's dominant. So it's what you're seeing. But the genotype is what is coded for. So a little bit of an example here of a few Punnett squares. We're not going to get too crazy into this, but I do think that it's important to have something to visualize the different crosses, not just between the 2 heterozygous examples. So the little key here is that our dominant color is going to be red, denoted with the capital A, and the recessive color is going to be the B, denoted by the lowercase a. So the first example we have is what happens when you cross a parent 1 being homozygous dominant and a parent 2 being homozygous recessive. So you put parent 1 on top, 1A on each square, because those are the 2 possibilities for that parent's gametes. It can either pass on a big A or it can pass on a big A. And then along that left side for the other portion of the square, you put the parent 2's possible alleles, in this case 2 lowercase blue a's. And then for each square, you bring the letter from parent 1 and the letter from parent 2. So parent 1 comes down from the top, parent 2 comes across. So as you're reading the diagram, it's where those alleles kind of meet up in the square is how you fill it in. So in this case, all of the offspring are going to be heterozygous. They're all going to end up being red. Remember, red is dominant to blue. So in this case, the phenotypic ratio would be 100% red. So it'd be 4 out of 4 red. But the genotypic ratio would also be 100% heterozygous. So 4 out of 4 or heterozygous, which makes sense. Parent 1 could only pass on a dominant allele, and parent 2 could only pass on a recessive allele. The next example we have here is, what if parent 2, instead of being homozygous recessive, what if parent 2 is actually heterozygous? So again, parent 1, we have on top 2 dominant alleles. Parent 2, this time we have 1 dominant allele and 1 recessive allele. Filling out the chart across and down, we see that on top, parent 2 gave that dominant allele. You end up with 2 homozygous dominant individuals. On the bottom, parent 2 had that recessive allele. You end up with 2 heterozygous individuals. Now, like the last example, all of the offspring are going to be red. That is the dominant allele. Everyone has that dominant allele. But instead of the genotype matching the phenotype and having the same ratio, we're going to get half are going to have that homozygous dominant and half are going to be heterozygous. So it's going to be a 50-50 ratio here. So, and then moving on to the next example, if parent 2 is the heterozygous, or excuse me, the homozygous recessive, and parent 1 is the heterozygous individual. So filling out the Punnett square along that left side where parent 2's alleles are put, they're both the same lowercase a, denoting the recessive trait. On top, we see that parent 1 now has 1 dominant allele and 1 recessive allele. So when we fill out the chart now, under the column for that dominant allele, both of the offspring are going to end up being heterozygous. But they're both going to be red because red is the dominant trait and they have that dominant allele present. The other column, you'll notice both of the offspring are going to be homozygous recessive because both parents had that recessive allele to pass on in that case, in that column. So both of the offspring are going to be homozygous recessive. Both of the offspring are going to be blue. So the phenotypic ratio is going to be 50% red, 50% blue, because you have the 2 heterozygous offspring and the 2 homozygous offspring. But in this case, it is homozygous recessive. And again, if you have the homozygous recessive, you get that recessive trait expressed. Whereas in the heterozygous case, the red is going to cover up the blue. And also in this case, we get the genotypic ratio that is going to be the same as the phenotypic ratio. And then our last example, which is exactly the same as the pea plants for the most part. Both parents are heterozygous, and you get that 3 red offspring, 1 blue offspring, but your genotypic ratio is going to be 1 to 2 to 1. So I know that's a whole lot to take in. I know that ratios can be really confusing, but when you're doing them, it's just counting. That's the important thing. When you're learning about Punnett squares, learn how to fill them out, learn how to take the allele from the top, bring it down, do the same on the next one, take that second allele, bring it down that column, and then move on to parent 2. Take that top allele, move it across, and then move down to the second allele, move that one across, and then step back, look at what the information's telling you, look at what each square is saying. First, figure out what you're going to see, figure out that ratio by counting. It's just counting, I promise. So in the cases where you have 4, you have how many out of 4 are red, how many out of 4 are blue. And then look at what the letters say. How many out of 4 are homozygous dominant, how many out of 4 are heterozygous, and how many out of 4 are homozygous recessive. Just take it one step at a time, and you can do it. Punnett squares look confusing at first, especially when you start getting into the ones that you're looking at more than one trait. We're not going to look at any of those today. We will look at those in the next video. So for now, this is all I have for you. Please leave any comments if you need clarification on anything down in the bottom of this video. Let me know if there's any more study material you would like. I'm probably going to take those Punnett square slides and remove the insides of the squares and things like that and include them in a PDF to go along with this video because it's good practice to have. So until next time, if you're struggling with this Punnett square thing, take it one step at a time. That's going to be the easiest way to get the hang of it. And don't move on to the big scary Punnett squares until you have these very basic ones down. Because you got this, you can do it. Genetics is not as scary as it looks. So have a wonderful rest of your day, afternoon, evening, what have you. I will see you all in the next one. Please don't forget to like, comment, and subscribe, and follow me on all my social medias. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts.
If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane V. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.